record on this computer for now. Great, so if you're just joining the recording, we've just gone through this outline to say we're going to talk about how top-down data differ from bottom-up data, how we can separate proteins efficiently, because separation is the key to giving our mass spectrometers a lot of different opportunities to look at ions in the mixes. We're going to spend a lot of time on applications of top-down proteomics, and finally talk about what happens if we alter the boundaries between top-down technologies and others. All right, so let's start with the fact that top-down tandem mass spectrometry is going to differ substantially from the data sets that we produce in bottom-up mass spectrometry. So in bottom-up mass spectrometry, we may use a data-dependent mode where we say what ions representing peptides are present in a mass spectrum, and then targeting the most intense of those for interrogation by tandem mass spectrometry, by producing the fragments from a particular set of peptides, and then producing a scan of the fragment ions produced when we blew apart this peptide. However, there are a lot of differences when what you're fragmenting is a protein rather than a peptide. You know, most of the peptides we deal with in shotgun proteomics are not bigger than, say, 20 amino acids typically. You'll certainly see some that are larger than that, but generally our wheelhouse is somewhere between 12 and 25 amino acids, perhaps. In a top-down tandem mass spectrum, however, you're not producing just fragments from a very short peptide, but rather from the entire proteoform in its entirety. So this means that we're going to have a lot more fragments to deal with. If you have a 20 amino acid peptide, there are 19 peptide bonds conjoining them, and shattering any one of those peptide bonds is going to produce an N-terminal fragment and a C-terminal fragment. But if you have 50 amino acids, you have 49 peptide bonds, and each of those 49 breakpoints can yield its own pair of fragment ions. That's quite a lot more information jam-packed into the spectrum than what we would have in a tandem mass spectrum of a peptide. Similarly, we are accustomed to having shotgun proteomics focusing on +2 and +3 charged peptide ions, the precursor ions. And that means that when you break apart a +2, you're likely to produce a +1 and a +1 fragment. If you break apart a +3, you're going to get +1 and +2 charged fragments. But what do you get when you're working with a protein ion, an intact protein as an ion that has 50 amino acids? Are you able to get up to 7 charges, 8 charges, 20 charges? Large proteins can take on very large charges, and when you break them to produce fragments, those fragments also have high charge states. In fact, it's frequently the case that the fragments produced from an intact protein will appear in multiple charge states. So I've already said that our tandem mass spectrum is going to be crowded because we've got lots of peptide bonds. When we add to this the problem of having huge numbers of protons charging the peptide, we have an even more crowded tandem mass spectrum because the fragments appear at multiple charge states. It's also the case that when you have a more massive peptide ion, it's not likely that the isotopes you see of it are just the monoisotope and maybe 1 or 2 other isotopes. When you have something that's got 50 amino acids in it, you're going to have lots of isotopic variants that appear for it. And finally, there's the problematic relationship of fragmentation technique with proteins. If you're using a technique like collision-induced dissociation, you generally don't get a very complete series of fragment ions that can show you the sequence throughout the protein sequence. So we've often seen that people who are dealing with very large ions that take on large charge is that they use a technique called electron transfer dissociation, ETD, which causes, as you can see at the bottom right in this slide, instead of having a breakage at a peptide bond, like you would see in most shotgun proteomics that produces a B-Y pair of ions from the breaking peptide, we see that our breakages are no longer at the peptide bond, now they're between the nitrogen and the alpha carbon. And it's produced by a very different kind of process. Instead of basically barraging the peptide with gas collisions until it breaks apart, not a very gentle process, we are instead causing the protein ion to capture an electron, which creates a radical and causes a destabilization of these bonds. So that's a relatively gentle process. But people have shown that in many, many cases, an intact protein may accept an electron and yet not break apart into a C-Z ion pair. As a result, you'll sometimes see that we use ETD, but we also tickle the protein a little bit. So we add a little bit of collision energy that causes the fragments to separate from ETD. The offshoot of that, though, is that instead of just producing a C-Z pair for each peptide bond, well, for each amino acid, I should say, but you also see some amount of CID style fragmentation. So in effect, we have fragments that are more numerous because of the peptide bonds. We have more charge variants of those fragments present. We have more isotopes because these are more massive fragment ions. And we've got both BY pairs and CZ pairs, making these spectra very complicated. Now, if you're familiar with analytical chemistry, I think what's coming next should not be a surprise to you, which is to say that although the mass spectrometry community has become very fixated on accurate mass, knowing to the third decimal place what the exact mass of a fragment ion or a peptide ion is, in this case, we have a problem associated with resolution, which is to say that multiple fragments may appear very, very close together in the spectrum. And being able to distinguish this peak from the next becomes a much more challenging task when the spectrum is this crowded with fragments. Okay, now there are three kinds of visualizations that I think might seem a little unfamiliar if you're accustomed to looking at only shotgun proteomics data. So I'm going to try to walk through these. You'll note that I've used the abbreviation PRSM at the top. This is to say that instead of looking at peptides, we're now concerned with proteoforms. A proteoform is a particular variant of a protein sequence along with any modifications it's carrying. So it might be that the protein sequence is clipped. It might be that it's carrying methylations and oxidations and phosphorylations. That particular configuration of clipping and of sequence variants and of PTMs, post-translational modifications, constitutes a proteoform. So an individual protein coding gene may generate multiple proteoforms that we want to measure. They get represented in these three different ways. Okay, so we start at the lower left here with isotopic envelopes. I want to stress that we are looking at a mass spectrum here, which is to say that we haven't fragmented anything in this case. We're just looking at the mass spectrum to see which protein, or specifically proteoform, ions have been detected. We see that there's a whole scattershot of peaks all in an envelope together like this. This is something that we call an isotopic envelope. Remember that any carbon that you see in a protein is generally carbon-12, but about 1% of the time, a carbon is instead a C13, or a 13C, a carbon nucleus that has one additional neutron to it. As a result, when you have a very large proteoform, you're going to see many isotopic variants, in some cases 5, 6, or 10 extra neutrons that are riding around in this nucleus. As a result, the signal for one proteoform is separated across multiple isotopes. In this case, I note that this packet of isotopes is separated from the same protein, H2B2 here. You see it's H2B2 here and H2B2 here. These differ by an acetylation. So acetylation is a post-translational modification that marks this protein. So we see that this isotopic envelope and this isotopic envelope are separate proteoforms, and yet they come from the same gene. Okay, so this is a way that we can represent top-down data, not looking at the fragments from them, but rather at their overall intensities as a function of different isotopes at a given mass to charge value. I want to note that this is not the full mass spectrum. We see that it starts at 830 m/z and runs up to 838 m/z. This is just an 8 m/z window, and yet we see that it's a huge number of isotopic envelopes all packed into this tiny space. The entire mass spectrum is going to show a huge number of proteoforms for us to compare. I'm going to skip over to the right edge of this slide, if I may. so good. Tea is just the life giver. Here we are looking at a tandem mass spectrum. So this image that we see at the bottom has a range from about, say, 700 m over Z up to about 4,000. And we see that it's just littered with different fragment ions. Over here, we're looking at intact proteoforms. Over here, we're looking at the fragments produced from a particular proteoform. Okay, so this type of spectrum looks like a very complex lawn. You wouldn't want to mow that. But if we zoom in on just a tiny, tiny little sliver of this tandem mass spectrum, we can see the information that reveals the sequence of the proteoform in these tandem mass spectra. So let's zoom in on this tiny little red peak and boom, look at all the stuff that's packed into this tiny space. Here we're looking at a 10 m over Z window, just from 2067 up to 2077, and we see it is positively littered with fragment ions. I wanted to point to some of the things that we mentioned in the previous slide. Here we have a doubly charged Z ion, so it represents the C terminus after a breakpoint. We see that 1, 2, 3, 4, 5, 6, 7 peaks with a little black ball above it appear. That is to say that we have that many isotopes present for the Z37 ion containing the last 37 amino acids, charged at +2. We know that it's a +2 fragment because these peaks, these isotopes, are spaced 1/2 m over Z from each other. Now, that's a much simpler case than when we get to something like this C80 ion. So here, this is the N-terminal ion from ETD. It contains the first 80 amino acids, and it's quadruply charged. So we see that where the Z37 ion at +2 had its peaks spaced every 0.5, the C80 ion has its isotopes spaced every 1/4 of an m over Z. It's the reciprocal of the charge state. So even though we're looking at a very tiny part of the tandem mass spectrum, it is absolutely littered with fragment ions appearing in different charge states, containing different number of amino acids, appearing in multiple isotopes. These spectra put a real premium on resolution. It should be apparent that looking at the annotations of of tandem mass spectra is quite complex when we get into top-down proteomics. And so I would note that one of the most common visualizations we see is not to simply mark the fragments in a tandem mass spectrum, as has been done at the right. Instead, you'll frequently see what's called a fragment map. This is based off of a nomenclature for how we name fragment ions that was introduced by Ropsdorf and Folman back in 1984. So at the top, we have the protein sequence running from the N-terminus down to the C-terminus. It's not marked here, but that's the C-terminus. And we see all these different amino acids that fall within this proteoform. And we observe that for some of them, an N-terminal fragment has been observed, and for some of them, a C-terminal fragment has been seen. So the N-terminal fragments are either B or C ions. The C-terminus containing fragments are the Z or Y ions. All right, so typically when someone is asking, how much faith should I have that this tandem mass spectrum has been correctly identified? I'm going to do it by visualizing it in a fragment map. You can see that for some of these ions, we have only evidence from the N-terminus. All of these that have a flag pointing off to the left but not to the right are cases where we have the N-terminal ion only. Ions down here that contain just a flag pointing to the right at the bottom, those contain only the C-terminus. And relatively few of the peptide bonds have evidence from both the N-terminal and C-terminal side. We do for this little AG bond. Up here we have a pretty good run of fragment information that shows that all of these had both termini represented for each of these peptide bonds. So a lot of the time, instead of showing marked up tandem mass spectra like this, you will instead see in top-down proteomics people substituting fragment maps. All right. So I'd like to talk about a whole lot of different ways that we can separate proteins from each other, but I want to try to group them together in a way that helps us to keep these things organized. I would like to start with this concept of separating on the basis of size. I think a lot of people here have worked with 1D gels at some point. You've tried to separate proteins by sedimenting them into a gel. And the little ones move this speed, the big ones move that speed, etc. So we see that if you do your separations in a normal gel, one of the big problems you're going to have in top-down proteomics is being able to get enough protein out of the gel fragments or the gel bands to be able to analyze them on the mass spec. So some folks have worked on this technology called gel-free down here at the bottom that allows us to separate proteins through gels, but they elute in the end into fractions. They run out of gel space essentially and go ahead to drip into the fraction. So gel-free is a way that we have better recovery by just electrophoretically moving them all the way through the gel and into different fractions downstream. Those can then be analyzed by something like reverse phase liquid chromatography with really short beads. Carbon 4 rather than carbon 18 would be kind of the norm for this. The reason that the beads that we typically use in bottom-up proteomics are really hydrophobic. They're 18 carbon chains, and proteins stick to those so well that we can't really recover the proteins once we've got them stuck to it. So typically, if you're doing reverse phase with proteins, you're going to use much shorter hydrocarbon chains on your beads. Okay, so that's one. Gel-free is a way to improve the recovery of protein from a gel separation. You may be familiar with LC. Talking about reverse phase liquid chromatography is one kind of liquid chromatography, but there's also one called size exclusion chromatography that allows us to impose a cut basically on which proteins are above or below a hydrodynamic radius. So we're separating them in LC rather than in a gel per se. And of course, you can also separate proteins by flowing them through gas. So a really large ion that has a large collisional cross-section is going to be slowed by lots and lots of collisions with gas as it moves through this chamber, whereas a relatively small ion takes on relatively few collisions and moves more rapidly in ion mobility. So there are, the company that really staked its flag in the turf first in the space of ion mobility was Waters. But these days, there are a lot of different mass specs being sold with ion mobility sources on them. So I would note that timsTOFs have begun becoming very popular in the proteomics community from the company Bruker. And I know that Agilent, for example, has ion mobility. So there are lots of different companies now that are starting to move into the space of allowing for separations on collisional cross-section, which is related to size, in ion mobility chambers. So this has become a really popular option for doing that. And you can see up at the top, I've included a diagram that shows one of these ion mobility chambers for separating ions on this cross-section. Okay, next, charge and pH-based separations. I think that many people have run 2D gels, and one of the things that you do to run a 2D gel is to first separate the proteins by their isoelectric points. This takes place in an isoelectric focusing strip. So you can use this for starting a top-down experiment, and it's a very established technology. Again, there's a little bit of concern about whether you can adequately elute proteins from these strips once you've done the separation for downstream mass spectrometry. So one of the things that's become popular in top-down proteomics in particular is something called capillary zone electrophoresis. This has often been substituted now for reverse phase liquid chromatography. So whereas in shotgun proteomics, RPLC is almost universally used as the stage that gets you into electrospray, in top-down, it's got enough problems that people have really tried to tinker out different methods that could substitute for it. So down here at the bottom, I've included an image that was provided by Eli McCool, who wrote this paper, actually. It was very kind of him to provide that. You see that here we have the Electrospray ionization source. This is what's going to jolt the proteins into the gas phase as ions. And we also have coupled to it, not reverse phase liquid chromatography, but rather capillary zone electrophoresis. So this is a type of liquid separation, but the solvents are not moving through these columns to force the ions to move. They are moving in response to electrical separation. So a very high charge can cause the ions of proteins to move toward the electrospray ionization source. Capillary zone electrophoresis has some really big pluses and some really big minuses. The really big pluses are that because you're doing this in solution, you can use a pretty high voltage, and the heat comes off of the column pretty well, and you get a very rapid separation of ions based on their charge essentially. So if you have the pH at which you're running it is going to determine what charge states individual proteins take on, and you can do a separation really quickly with a huge number of plates of separation, so very clean separations among different proteins. The trouble is that capillary zone electrophoresis doesn't let you pack a huge load of protein onto the column, and that's been something that we've struggled with for a long time. But we see that in top-down, it's made its reappearance and can be a very valuable replacement for liquid chromatography. And of course, if you're familiar with techniques like mud pit from shotgun proteomics, you can see that something like this happens within the space of top-down. Strong anion exchange and other types of ion exchange chromatography can be used for fractionating a very complex mixture of proteins into different fractions. Those fractions may then be analyzed by reverse phase liquid chromatography, again, using shorter chains than you would in shotgun proteomics. I want to note that I personally find the second and third types of separations on this slide to be very, very easy to confuse. If you've heard of hydrophobic interaction chromatography, you may also have heard of hydrophilic interaction chromatography. These are obviously working from the opposite point of view. One is checking for interactions with very non-polar parts of the protein. The other one is interacting with the parts of the protein that are really very, very fond of solution type, of acidic and basic sites on proteins in particular. Okay, they have the same three letters starting their names: hydrophobic interaction chromatography, hydrophilic interaction chromatography, and yet one of them goes by the name HIC, hydrophobic interaction chromatography, and the other one goes under the abbreviation HILIC. So if you see HILIC, you can always remind yourself that it rhymes with hydrophilic. I hope that helps. So we have different ways that we can do these separations, and by making these mixtures separate into different protein collections, we have a much better shot of separating them well when it comes to introducing them to the instrument in reverse phase liquid chromatography. Okay, we've already covered a lot of ground in analytical chemistry, but from here I'd really like to move to use cases. What are the applications of top-down proteomics that have convinced people to labor for years popularizing and enabling this, this type of analysis by creating new technologies? This has been the coming thing for my entire career. So I think it's worth asking, what biological drives cause people to need this technology, to work so hard to bring it into fruition? So use case number one: what combination of post-translational modifications enables biological activity? If I have a protein of 50 amino acids, and that's not large, just 50 amino acids, I might see that in its translated form, this protein takes on basically no biological activity. But if I add a couple phosphorylations to it, suddenly it's as active as anything. So in a case like that, it may be that phosphorylating this point or that point has an equal effect. But more generally, we expect that different modification sites associate with different kinds of switches, right? Just because if you're in a science fiction television show and you walk up to an alien control panel, you always mysteriously know which switch to flip to turn on the engines. But that's not how biology works. In biology, discerning which switches, which PTM sites are associated with activity is rather challenging. And we note that different combinations are not independent of each other. That a phosphorylation at site serine 50 versus one at threonine 48 may have interacting capabilities, that both of them need to be phosphorylated, or it's just as though neither of them is phosphorylated. So if you're trying to do this kind of analysis by shotgun proteomics, you're going to be limited to the fact that individual peptides may come from different proteoforms. If different proteoforms have been phosphorylated differently and you produce shotgun peptides from different proteoforms, you might say, well, we know that this site and this site and this site are phosphorylated, but that is a wrong claim unless you have one peptide that shows all three of these have been modified on the same sequence, right? So let's talk about histones. If you're interested in epigenetics at all, understanding the drivers behind gene expression regulation, then you probably care quite a lot about histones because their modifications have a lot to do with what kind of transcription is taking place around them. Here I'm showing the sequence for human histone H3.3. Now you'll note that I've marked every arginine and lysine in the sequence in red. And the reason for this is that if you use trypsin on histone 3.3, those red amino acids are your indication of where trypsin is likely to cut. So if you were to digest up this protein in trypsin, you would see that you get a whole lot of very, very short peptides. I mean, look, this is 1, 2, 3, 4, 5, 6, 7, and that's one of the longer peptides, plus this really long one starting with F, Q, S, A, I, etc., down to this KR. So you get one long peptide and the rest of it is basically sliced down to tiny little slivers of peptide. Using trypsin on this guy is not a very, very good idea. All right, now I would note that Gary et al. Published in 2020 this really interesting interrogation of histone 3.3, and you can see that they are asking about comparing versus controls, a mutant versus a wild type, which modifications were changing in this sequence when this was seen in control samples or when it was seen in the mutation site. So here we see in a mutation variant, here we see kind of a complex nomenclature, and I want to try to break this down because it shows up a lot. Here we're looking at lysine at position 4 in the sequence, this one right here, in other words, and we're asking about a single methylated form of that. What we're noting here is that the lysine is not just a trypsin cutting site, it is also a location where it's possible for a methylation to take place. In fact, that's not the only possible modification that can take place at lysine 4. Lysine 4 might be singly methylated, or it might be trimethylated. Now, if you remember your organic chemistry, you probably think to yourself, now a methylation adds 14 mass units to this site. A trimethylation is going to add 3 times as much, which is about 42 mass units. But I want to note that we're also paying close attention to the presence of acetylation, and acetylation also has a mass of 42. So we need to be able to resolve very close masses to be able to discern a trimethylation from an acetylation. And in fact, at the lysine at position 27, we must distinguish between trimethylation and acetylation because they behave differently in this mutant versus wild type format. So there's a lot of detective work that can be done to ask about the conjoint set of modification states, to be able to say these modifications all take place on the same proteoform. You're not going to get there with shotgun proteomics. Top-down is needed. So can we use post-translational modification data from top-down to separate different proteoforms? I wanted to point to a result from a group at Pasteur Institute in France. Here they are looking at an archaea. So this is not a bacterium. It's not a photosynthetic microscopic organism. This is an archaean that's been found in some places very far north. They have chromatin proteins, which is a little confusing. I mean, some people would even call the structures that archaea use for manipulating their DNA histones. They just like you or I carry. So here we see then that we've got 5 different proteins that are all known to interact with the DNA in this bacterium. but they note a confusing, complex set of PTM variants, which each represent different proteoforms. So in this first case, we see that we have an acetylation appearing in all cases at the N-terminus, but we see that this lysine showing up on the first row shows up with a methylation in this proteoform, a dimethylation in this proteoform, sometimes with a dimethylation and an oxidation. Now, I note that we can't really tell in most cases whether an oxidation happened in vivo or happened when we were working with a sample on the bench. But in some cases, the same residue may be found in more than two, in this case, three different modification states: singly methylated, dimethylated, trimethylated. We see that methylation is a pretty common regulator, in fact, on many of these different proteoforms that we see. So certainly, different proteoforms may be observed that are differentiable on the basis of PTMs. In this study of sarcopenia, does anyone know what that is? Sarcopenia is kind of a reduction in muscle mass that occurs sometimes in connection with aging. Here we see that the proteins tropomyosin and myosin light chain are being interrogated. So this is the tropomyosin side. This is the myosin light chain side. Here we see that alpha tropomyosin shows up in two different places. Here we have the normal form. Here we have it after it's been phosphorylated. The beta tropomyosin is present in an unmodified form and in a phosphoform. So we're able to ask from the 6-month to the 24-month to the 36-month variants, cohorts that we're looking at, we see a different amount of phosphorylation occurring in some of these cases. All right, so here we're looking at the unmodified form of the myosin light chain. We see that there's a huge amount of its phosphoform. At 36 months, however, the unmodified form is almost as intense as the phosphoform. Okay, so yes, PTMs can be separating different proteoforms. We can even quantify to some extent how much a given PTM is changing in ratio among different cohorts. All right, so that's a good bit about PTMs. Let's also talk about which of several transcripts may be translated for this gene. I would start by saying that most people doing bottom-up proteomics have kind of given up on their ability to discern different proteoforms based on which transcript is being produced. This is because in many cases, people who download reference proteomes for use in identifying tandem mass spectra only download one isoform, one translated transcript to represent each gene. As a result, their sequence databases don't contain all the different isoforms that could be detected. They include only the longest, typically, if you're grabbing it from SwissProt. So in a lot of cases, you'll find that bottom-up proteomics people say, I'm willing to say this gene is being expressed, but I'm not going to keep digging into this to say which transcript in particular from it is translated. So I thought it would be worthwhile to point out the research of Wei et al., who were working with a gene called PDLIM7. And you can see up in A, the set of exons that are associated with this gene model. So we have an untranslated region that appears in exon 1. Much of exon 15 is also untranslated. Much of exon 9 is not translated either. So we see that kind of the longest isoform that we can produce from it contains all but, let me see, here we're missing the 6th exon and we're missing the 9th exon, but I think all of the others are present. That's the longest isoform. In some cases, we see that exon 6 is included, but not exon 5. You see that? So if you're trying to discern which of these exon splice events have taken place, you really need to have a peptide that lies right down on the intersection between two exons, so that you have a few amino acids in this exon and a few amino acids coming from this exon, and therefore you can say, surely the isoform that produced these has spliced these two exons together. But does that help you discern all of these different transcripts that may have been produced? In this case, They were able to detect a novel isoform that it contained a splice collection that had not been anticipated from just transcriptomics by using top-down proteomics. And that's very powerful. A top-down proteoform is almost always going to span multiple exon-exon junctions within the gene model. And that's going to give you a lot more power to say which transcript relates to this particular proteoform. Certainly, there's a lot of protein modification that involves clipping off particular amino acids. I think many people know that in lots of cases, we find that the first amino acid of a polypeptide is methionine, and in many circumstances that gets truncated. So the first methionine gets chopped off of the sequence. You can see an example of that appearing right here. I think a lot of people are also aware of pro-peptides. For example, when your body produces pepsin, it actually produces pro-pepsin, and it's only when that pro-peptide gets clipped off that the protein swings into action. Similarly, our bodies frequently make use of signal peptides to represent a kind of shipping label on a protein. So the protein is manufactured, the cell machinery reads the label and says, oh, you're a cell surface protein, and packages it out there where that section can be lopped off for the protein. Basically, the shipping label gets taken off so the protein can swing into action. But how would you measure this in shotgun proteomics? If you were trying to discern whether this was or was not clipped off in the final version of the protein sequence, yes, you can digest it up and identify peptides, but you may find that you have very few peptides covering this particular protein sequence, and therefore you don't really know whether you didn't see this N-terminal part because it wasn't there or because you didn't sample enough peptides there. We don't really know. If you're measuring the proteoform in intact form, you have a much better shot of being able to discern which parts of the protein sequence are lopped off before the final version of that protein is present. So Chen et al. Were using this in the context of mouse MDSC cells. And here are the different proteins that they were looking at. You see that they've got many different red arrows on this histone H2A. That reflects the fact that some of the proteoforms started here, some of the proteoforms started here, others started here, others started there. There were lots of different clips of this histone that were present in the sample. Okay, very big deal. Now, I think a lot of people are thinking a lot about SARS-CoV-2, the virus that causes COVID-19. I just want to point out that many viruses use a really strange strategy if all you've ever looked at is prokaryotes and eukaryotes. When you look at viruses for the first time, you can ask, how could that possibly work? So if you've looked at the annotated genome of SARS-CoV-2, you see that there's this massive region called ORF1A, a smaller region called ORF1B, and then you've got this other stuff back here. So when ORF1A is produced, it doesn't produce one protein. It creates something called a polyprotein. You can see then that it contains a whole bunch of short sequences that need to be released from the polyprotein before they can swing into action. So we see that two different proteases, a papain-like proteinase called PL and a 3CL protease, have to cut into this ORF1A and ORF1B to release the mature polypeptides to swing into action. But there's a chicken and egg problem, because we see that the proteinase itself is encoded by this ORF1A. So when SARS-CoV-2 infects a cell, it had better bring along more than just its RNA. It must also bring along the proteinase, some copies of the proteinase, to first chop those first translated sequences apart, so that more proteinase can be produced, and more of these polypeptides can be released into the cell. Okay, so in this case, if you were looking at the proteome of SARS-CoV-2 with shotgun proteomics, you would not be able to discern the extent to which the proteinase had been able to cut up these ORFs. Okay. I think I'm allergic to something in the springtime here, but my vocal cords gotta get dry fast. All right, so can we in fact do quantification, not just detection of things, but quantification of things in top-down? I'd like to start with this paper introducing something called NuCode. It's a cool name, I think. I mean, Nucode, Silac, iTrack, we've got all these different brand names now, but Nucode was a clever name because it's a neutron encoding strategy. This was released by Josh Kuhn's lab by somebody named T. Rhodes here. He created a strategy by which lysines were present in labeled form. So here you have to be able to grow your samples in culture in order to be able to label them properly. So he's got a lysine that he's calling in this diagram 602, and a lysine in this that he's calling 080. The coding is a little squirrely here, so I'm going to try to unpack that for you. The 602 is a code for CHO, like you see in carbohydrates. So the 6 refers to 6 labeled carbons, 0 labeled hydrogens, and 2, oh, I'm sorry, that's actually a nitrogen, I think. Sorry, CHN. Okay, in this case, the lysine has been labeled with 8 heavy hydrogens. Okay, so you might ask yourself, if you're adding 8 mass units to the red lysine, and you're adding 8 mass units to the blue lysine, aren't they going to show up right on top of each other in a tandem mass spectrum? And the answer is no. They have different energies of adding in the neutrons in hydrogen versus the other elements. As a result, in this case, we are looking at a intact peptide that's charged +9, and our range here is only from 569.4 to 570.4. In other words, from here to here is a single m/z unit. Within that, we have an isotopic packet where the isotopes are spaced every 1/9 of an m/z. And because the instruments we can use today have such amazing ability at resolution, we are able to distinguish not only one isotope from the next, but the light 602 lysines from the heavy hydrogen labeled lysines. I think that's kind of astonishing, frankly. But in this case, we know that this intensity can be compared to that intensity. These are very, very closely paired. And they did a nice experiment where they mixed together samples in known ratios to ask whether Nucode was giving back correct ratios. Here they had mixed together 2 samples 1-to-1. Their actual estimation based on the data was that they were at a ratio of 1.05, so 5% away from truth. Here we had a 3-to-1 mixture of red to blue. Here we see that the empirical measurement was a ratio of 3.15. They did a mixture of 5-to-1. The more extreme the measurement, the harder it is to get that right. And their Nucode was able to produce a ratio of 4.5. I think that is that's a pretty strong testament. Now, can you do that for all ions? Are all ions going to be ratioed so closely as that? Probably not once you get down into the grass, but at least for some of the proteins, you're going to have the ability to do a very careful quantitation of how much difference there is in them. And as we mentioned previously, you may also want to ask about how much you're shifting from one proteoform to another. So here developing ratios in that space. Here we have a protein ZEO1 that starts with its unmodified sequence, its 1 acetylation sequence, and its 1 acetylation and 1 phosphorylation sequence. Why are these two so close together? Acetylation adds 42 mass units, and then when you add the phosphorylation, you add about 80 mass units. So these are twice as far apart because it's about twice as massive. Okay, what we see then is that when, in the case of Entai et al., when they deleted the gene for histone deacetylase, what does histone deacetylase do? It removes acetylations from histones. Clever, right? Great naming strategy. So here we see that in this case, histone H4 in wild type circumstances has 1 acetylation at this level, 2 acetylations at this level, 3 acetylations here, 4 acetylations there. When the deacetylase is removed, however, this singly acetylated form goes away altogether. It just vanishes. So the deacetylase is obviously doing some work. When you knock it out, you see a higher cohort of a higher degree of acetylation observed on histone proteins. That is a quantitative story told with PTMs. All right. I see the time here. I want to be sensitive to everybody's schedule, so I'm going to move ahead as quickly as I can. Antibody research is obviously very important. Anyone who's been following COVID-19 knows that the Regeneron treatment of injecting antibodies into patients with COVID-19 helps them to respond more effectively, we think, to the COVID-19 virus. In this case, 20 laboratories were given 3 monoclonal antibodies, so not a mixture of antibodies, just single antibodies at a time, and they were using Fourier transform instruments, Orbitraps, and time-of-flight instruments. That's an important thing. You don't have to have Fourier transform to be able to do effective top-down work. So in this case, we see that something called IDES was used. This is a papain-like protease. And if you think about an antibody, we often think of them as these big Ys, but if you're not an immunologist, you may lose track of the fact that this top part, the Fab fragment up here, these are the parts that are specific to a particular antigen. This stem down here is necessary for interacting with the rest of the immune system, but it's not very specific. It doesn't really have any binding affinity to the antigen. So if you were trying to discern how pure a particular antibody preparation was, you'd care a lot about this top part and not so much about the handle. So if you treat it with something like IDES or another papain-like protease, you can separate the Fab, the specific part, from the non-specific part, the fragment crystallizable part. I'm going to call it the handle. I hope you don't mind. And they were able to show that laboratories using all 3 of these technologies, a very wide number of 20 different laboratories, lots of different instrument operators, lots of different instruments, were able to use all of these different platforms to come up with useful information about how heterogeneous a particular therapeutic antibody was. I think that's beautiful work. I'm always a big fan of these shared laboratory studies, shared sample studies where one sample goes to multiple laboratories and we get to play the results off of each other. It helps us to understand whether we as a community are able to use these technologies profitably. Now, as we wrap down, I want to remind you that your choice is not always going to be top-down or bottom-up. You may have the option of combining them in integrated top-down, bottom-up, as I've shown over here at the right, or you might use a technique called middle-down. So let's start with integrated top-down and bottom-up. In this case, we're showing isotopic envelopes for gamma-synuclein. Here we have a sequence variant, the glutamine, the glutamate has been replaced with valine here. In this case, we have the wild type sequence. So we see that these folks who are heterozygous have both of these different proteoforms present, here differing just by one amino acid being altered. Over at the right, we have kind of a complex diagram. So I want to note that this E110V tells us that this is amino acid 110. Here we see that the E has been replaced by a V. We see these little flags pointing left and right in some places show us the fragments produced when we shattered the proteoform in top-down proteomics. But we also have these yellow lines. These yellow lines represent parts of the protein sequence that were identified through shotgun proteomics. We have peptides that span across these entire yellow regions. So that's brilliant. We like having all that data. By being able to play them off of each other, we're able to ask how strongly we can support our identification of this particular E to V variant. Here we see that top-down did not give us fragment ions that would allow us to confirm that modification because the only fragments we had were very far away from it. But bottom-up proteomics was able to identify it so we could see it. Down below, we see that we have the wild type and another sequence variant here replacing an asparagine with a histidine. We see the histidine is over here, and in this case, we do have fragment ions near it. In this case, bottom up couldn't see it. There was no peptide that covered that histidine that was identified. Only through top-down proteomics were we able to detect that one. Okay, so pulling these 2 technologies together gives us more power than either alone. In middle-down proteomics, we step away from our battle horse. We love trypsin. Trypsin is our best friend in shotgun proteomics. And sometimes trypsin is not going to produce long enough peptides to be informative. Remember the case of the histone. So in a case like that, it's really valuable for us to look at other proteases that can break us into peptides that are not so very short as we see in trypsin. Lyse-C is a good example. And the naming of this tells us where it cuts. In the peptide sequence, if you've got a lysine, this will cut to the C-terminal side of it. Now wait, that must seem awfully familiar to you. Trypsin cuts at arginine and lysine, but Lyse-C doesn't cut at arginines. It will only cut at Lyse-C. As a result, you get much longer peptides. I mean, it's roughly double the distance to get to the next cutting site with Lyse-C as opposed to trypsin. We also have the ability to cut to the N-terminal side of aspartic acid. That's the letter D in a sequence. We also have the ability to cut to the C-terminal side of glutamic acid. That's an E in a sequence. And we see that the lengths of the peptides that result from them are quite different as a result. Now, I want to talk about something that seems really gonzo to me, but is actually just incredibly paradigm breaking. Native mass spectrometry gives us the ability to examine not just individual proteins, but protein complexes in mass spectrometry. So I thought this quote from Lenny and Heck was really good. The term native in native MS describes the biological status of the analytes in solution prior to the ionization event. So here we're looking at these membrane pore proteins. We see that the whole thing we're going to call a yellow hexagon. It might be that this blue part breaks off, that's the blue box, and it might be that you get the other part of it minus the blue box. Here we're going to call that a red circle. We see that all of these different features are visible as different peaks in this native mass spectrum. Now, you'll remember that I mentioned the value of ion mobility for separating ions based on their collisional cross-section. You can use that not just for proteins, but for protein complexes. Brandon Ruotolo published a really cool paper back in 2008 showing the ability to, if you haven't seen a diagram like this, I would just point out that this is a mass to charge axis, and this is a collisional cross-section or drift time axis. In other words, things that, imagine a dude with a parachute, right? If he's got it all strapped to his back and he jumps out of an airplane, he falls really fast. That dude has the same mass, though, as somebody who has opened the parachute, right? So the cross-section of the parachutist with the parachute open is much bigger than the parachutist who's just dropping like a rock. In the same way, a protein that is loosely folded up is going to be a bit of a parachute as opposed to one that's all folded up like a baseball. Okay, so where they appear on the y-axis is that. Now you might be saying, this doesn't even look like a mass spectrum to me, and that's because you're probably looking for the intensity axis. In this case, it's coming out toward you from that. So a more darkly colored dot represents an intense signal, and one that's pretty low and gray is something that's close to the grass. Okay, so that's the very short guide of how to interpret ion mobility spectra. Lots of takeaways here. I appreciate your sticking with me all hour on this subject. And we do have, I think, a half hour for questions after this. So if you need to rush away, I understand. Top-down proteomics evaluates at the level of proteoform rather than the peptide. This is really important because we do not need to use protein inference or protein assembly to say which proteins have we observed given that we've seen these peptides. At the level of proteoform, you know exactly what the intact form looks like because that's what you analyzed. Enough software tools now exist to allow biological questions, not just technical ones, to be asked of our data. That's very powerful. This is kind of what's needed for this technology to make it out of specialist laboratories and into people who are trying to just learn some biology. Different preparations, different separations, different fragmentations are all likely to accompany a shift to top-down experiments. You shouldn't expect that today you use the instrument for a shotgun and tomorrow you use it for a top-down and then you switch it right back. You're probably going to need different pumps, you're probably going to need different kinds of columns, etc., for being able to use the instruments in this way. Biologically, top-down is very useful. which post-translational modifications co-occur across a proteoform? Which of several transcripts for this gene is expressed under these conditions? Are all parts of the sequence present in its mature form? Does the expression of proteoforms change under various conditions? How pure is this protein preparation? And finally, is this protein part of a larger molecular machine? These are really important biological problems, biological challenges that creep up in a huge number of categories. And in most of these cases, shotgun proteomics has important drawbacks that prevent us from using it for that. Okay, those are the slides for today. And I believe that the link has been sent to each of you to be able to download this PDF if you want to be able to refer back and find some slides. But I think, Tabo, that we are ready for some questions.
Yes, you can go ahead.
Tabo, are the students able to type questions into chat, or do they need to say them out loud?
They can do both. So maybe they can raise their hand and then we can acknowledge them and take the questions.
Okay. There was a lot of content today, so I know that it's a complex topic to talk about. But easy questions or hard questions are all okay. Tell you what, we'll even open it up a little bit more. If you have questions about careers in science, I'm also willing to answer that.
Can we just talk? I'm trying to type here. This is Genevieve here. Came in a little bit late. I just want to, I don't know if I followed correctly, but can you also do quantitative analysis with top-down proteomics?
You can. Yes, I talked about that in this slide on section 4. It is possible to use isotopic labeling, such as the new code strategy that's been shown here, to be able to ask, is the cohort represented by the red stable isotope versus the blue stable isotope more or less abundant? So yes, doing quantification is feasible in this context. It changes a fair bit, but it is it tends to be something that we do at the MS level rather than the MSMS level, but it is feasible.
So that would be relative quantification, am I correct?
That's correct. Yes. I would say though that almost all quantitation used in mass spectrometry is relative. You may be spiking in some number of some amount of a known amount that you're comparing against, but you're still computing it as a ratio for your experimental. So relative quantitation is almost always the basis for doing this in mass spec. Good question. Duncan, I see that your microphone. Oh, sorry. You don't have. It's a. I thought I was seeing a microphone was live. It's not. Other questions people would like to ask? Oh, Johnson, I see that you have your hand up.
Yes, I do. Can you hear me?
Johnson, yes, we can hear you.
Okay, yes. Thank you so much for the explanation that you gave. I really enjoyed it.
Thank you.
But then, from a chemist perspective, I just want to ask this funny question. Is there a way we can apply this to food and natural product research? Is it possible?
Okay, well, natural product mass spectrometry is kind of a specialized area in a lot of respects. Generally, when we talk about top-down versus bottom-up, we're talking about proteins. But I would note that natural product mass spectrometry is hugely promising. A lot of work, for example, from Peter Dorstein at UCSD, San Diego in the States, has been really, really important, I think, in opening natural product mass spectrometry for broader use. There's a very large database of natural products called GMPS that's run out of University of California, San Diego. So that if you produce a tandem mass spectrum of a natural product and you're trying to discern its structure from that, you may be able to detect similarities to structures in that database for recognition. So while today's talk is largely about intact protein mass spectrometry, it is possible to put stuff that's not proteins into the instrument as well and get good information back. I would highlight the example of venom proteomics, because a lot of times we see, or things like antimicrobial peptides that's secreted by frogs, stuff like that. Sometimes those peptides are translated at a ribosome, but sometimes they're assembled completely differently and use non-standard amino acids. So for stuff like that, you really need as much support as you can get to interpret the spectra that come back. But natural product mass spectrometry is definitely growing as a real technology that can be used quite practically. It is growing.
Okay, thank you.
Good question.
Thank you.
Dave, just a comment from John on the chat. There is a webinar next week on natural product MS.
Perfect.
I think Moladi sent an email yesterday or 2 days ago, so people can access that.
Yeah.
And then there's a question from Duncan as well in the chat.
Oh, let me see if I have the participant panel open here. Okay. Ah, from Duncan. Okay. Let's see. So Duncan asks, how efficient is ionization when performing top-down? The complexity of the eluting sample is not known. Gosh, I would say that this is a point of some difficulty. I would say that if you don't know the complexity of the eluting sample, you're almost surely going to need fractionation to deal with that complexity. Oh, Johnson, I'm muting your microphone. I hope that helps. Sorry about that. Okay. Okay. So I think that ionization efficiency is less a problem when you have a larger molecule like this, because there are more ionizable sites on it. It's also the case that even though I've highlighted Electrospray as a front end to mass spectrometry, a lot of people trying to do top-down are doing it with time-of-flight instruments, and for them, using a MALDI ionization source, ion source would be preferable. So I'm not sure I have enough experimental background to pin that one down, but I do know that the top-down community tend to be a lot more varied in their approaches to how ions get introduced to the mass spec in the first place. And I saw that John Becker said, oh, this really highlights the need for proteomics collaborations in the country. Absolutely, absolutely. I have been really glad to get to work with people at WITS and at CSIR and at UCT and so on. None of us knows everything. As is highlighted by Duncan's question, my experience being in front of the instrument myself is very, very dated at this point and largely related to nano LC-ESIMS. So yeah, my hands-on experience in top-down data production is nearly negligible. But we have more people. We have plenty of people in this country who are ready to take on that kind of challenge. So that's the collaboration is the way that we as a community move forward. Duncan asked, would modifications affect this? Yes, I would fully expect so. As we look at this slide right here, we might conclude that, I think the slides are still showing up on screen. So in this case, you might conclude there's more of the double acetylated than of the triple acetylated or singly acetylated, and both of those are stronger than the quadruply acetylated. That's a really dangerous thing to say because ionization can change when you add modifications. That something like putting a phosphate on board may cause the ion to fly better than one that doesn't have as many phosphorylations. So adding a in a 2D gel, when you add phosphorylations to a protein, you tend to see it change position on isoelectric on the isoelectric focusing side of things. In the same way, you may see a different ionizability because of modifications. Yeah. Let me see. Molati has shared next week's webinar on natural products. Good call. Yes. All right. Other questions that people have? Well, I would like to say that moving to South Africa was about the best thing that ever happened to me. I moved here in late 2015 to start a 5-year contract at Stellenbosch University, and I have loved it. I've loved being in South Africa. So I'm very grateful to the way this community has welcomed me, and I appreciate it. Always will appreciate it.
Thanks, Dave. We really appreciate you too. We've enjoyed working with you and sharing dinners after long workshops. It's been great having you here, and we hope to work with you in the future as well, wherever the road leads.
Thank you. Last chance for any questions.
Don't see any more hands or questions. If I can just repeat what I said earlier when we started the webinar, that we do have a Proteomics interest group going. If people want to join, please email me. I see Sean has already emailed me, so I'll add him to the group. The group, like I said earlier, is not a formal society. It's just a group of people in the Proteomics community working together, sharing ideas, sharing solutions, and also basically to foster or to help people collaborate better in South Africa and maybe have other connections abroad as well. And we have David now, so I'm sure we'll have lots of connections abroad. I think we can ask one more last time for questions. And does anyone have a question or comment? Doesn't seem so. It's been a great talk, David. Thank you a lot. Thanks for setting time aside to do this webinar. I know you've been busy. You had a very busy month, or couple of months. Thank you for setting time to prepare this talk and actually have it. It's been great, and I'm sure everyone has enjoyed it. It's been an interesting talk. I see people in the comments are saying it's been an interesting talk, so they really appreciate your time. Thank you. And thanks everybody else for joining in. Thank you for staying. We really appreciate your time as well. If there's, I don't think there's anything else from my side, David.
All set.
Oh, good. All right, and then with that, maybe we can end the session. And if you have further questions, you're welcome to email us. We have my email address, and I'm sure you have David's as well. So we can stay in touch. Thanks, everyone. Bye.